Good morning, students. Welcome back. We are going to continue with the lesson number 11, Enhancement of Food Production. Already we have studied about multiple ovulation embryo transfer and dairy farm and poultry farm management from the animal husbandry topic. Now, the next topic from this lesson is apiculture or beekeeping. It is nothing but rearing of bee for production of wax and honey. So that is rearing of honey bees to obtain bee products like honey, wax, pollens, bee venom and bee glue, royal jelly. These are all products of the apiculture or products of the beekeeping. Most common species that can be reared is Apis indica, that is from Indian or Indian bee. Then you know that honey is food of high nutritive and medicinal value. It is rich in antioxidant also. You know the benefits of antioxidant. Antioxidants, they reduce or they control free radicals in the body. So bee wax is used to produce cosmetics and fish polishes. For successful beekeeping, knowledge of the nature and habits of bees, then selection of suitable location for keeping the beehives, catching and hiving of swarms, management of beehives during different seasons, handling and collection of honey and bee wax. Bees are the pollinators of many crop species such as sunflower, brassica, apple, pear. Keeping beehives in crop fields increases pollination and improves the yield. By maintaining these important points in mind and by following this, we can successfully manage artificial beehives. Fisheries. Fishery is a branch of applied biology which deals with the catching, processing, fish farming and marketing of fish and other useful aquatic animals such as prawns, lobsters, oysters, mussels and crabs. Fishery is of two types, freshwater fish and marine fish. So there are some examples given along with the pictures: katla, rohu, common carp. These are freshwater fishes, and in marine, hilsa, sardines, mackerel, pomfrets, Bombay duck. These are all examples of marine fishes. Some important economic importance of the fisheries. Fisheries provides income and employment to millions of fishermen among farmers. Fishermen and farmers. Aquaculture and pisciculture are the techniques to increase the production of aquatic plants and animals. In 10th standard, we have studied in the biotechnology lesson about the Blue Revolution. It is the development and flourishing of the fishery industry. There are some methods by which fish can be preserved and prevented by spoilage. So some methods like chilling, freezing, freeze drying, sun drying, smoke drying, salting and canning. So this topic may come for 2 marks to enlist the preservation methods of fish. There are some byproducts of the fish along with the commercial value. They are fish oil, fish meal, fertilizers, fish guano, fish glue and isinglass. These byproducts are used in paints, soaps, oils and medicines. So preservation method of fish, economic importance of the fish, commercial value and byproducts of the fish are given on page number 257. Just go through the topic. Next important topic of the lesson is microbes in human welfare. Biotechnology is the application of scientific and engineering principles for the processing of materials by biological agents to provide goods and service to humans for human welfare. We are going to discuss microbes in household or food products, microbes in industrial products, microbes in sewage treatment, microbes in biogas production, microbes as biocontrol agent, and microbes as biofertilizers. Microbes in household products. Lactobacillus or lactic acid bacteria. It converts milk to curd by producing acids that coagulate and partially digest the milk proteins. Fresh milk can be converted to curd by having some curd containing lactic acid bacteria. It also increases vitamin B12. In stomach, lactic acid bacteria or lactobacilli helps to check pathogens. Bacterial fermentation, that is anaerobic, the respiration type is anaerobic respiration, in dough is used to make food such as dosa, idli, etc. Puffed up appearance of dough is due to the production of carbon dioxide gas. Baker's yeast, Saccharomyces cerevisiae, is used to make bread by fermenting dough. So along with the dosa, idli and dhokla, microbes can be used as a source of major foods, like you know that some mushrooms and truffles are directly used as food. They belong to higher fungi. They produce large, fleshy, fruiting bodies which are edible. Fruiting bodies are sugar-free, fat-free, but rich in proteins, vitamins and minerals and amino acids. The food in the fruiting body is low calorie. In the dairy products, lactic acid bacteria or lactobacilli are used for the production of curd, yogurt, buttermilk and cheese. We have studied production of the cheese techniques in 10th standard. Role of microbes in industrial production. You know that during fermentation, variety of products like alcoholic beverages, organic acids, vitamins, growth hormones, enzymes, antibiotics are produced. Production of alcoholic beverages. Saccharomyces cerevisiae is used in the production of beverages by fermenting malted cereals and fruit juices to produce ethanol or ethyl alcohol. Wine and beer are produced without distillation. Whiskey, brandy, rum, gin, arak, etc. are produced by distillation of fermented broth. The diagram of tubular tower fermenter is very important, which is given on page number 260. All of you please practice that diagram properly. Before we learn about the production of antibiotics and production of vitamins, there is production of organic acid. So microbes are also used for the commercial and industrial production of certain organic acids. This compound can be produced directly from glucose, for example gluconic acid, or formed as end product from pyruvate or ethanol. Organic acids like citric acid, microbe used in that is Aspergillus niger. Then gluconic acid, again Aspergillus niger. Fumaric acid, Rhizopus aureus, and acetic acid, that is vinegar, Acetobacter aceti. The chart is given on page number 260. Please go through properly. Production of antibiotics. Antibiotics are probably the most important group of compounds synthesized by industrial microorganisms. Antibiotics. Antibiotics are used to treat plague, whooping cough, diphtheria, leprosy, and many more diseases. Penicillin is a one antibiotic, or it was first antibiotic which was discovered by Alexander Fleming. There is a one chart given on page number 261, antibiotic producing microbes. So chloromycetin is produced by Streptomyces venezuelae, erythromycin Streptomyces erythraeus, penicillin Penicillium chrysogenum, streptomycin Streptomyces griseus, then griseofulvin Penicillium griseofulvum, then bacitracin Bacillus licheniformis, and tetramycin Streptomyces aurifaciens. This chart may come for 2 marks to match the following. Production of enzymes. In living beings, enzymes play very important role in metabolic reactions, and therefore they are very essential for living things. There are some enzymes which are produced by and their microbial source. Same enzymes are given on page number 261, like invertase, Saccharomyces cerevisiae, then pectinase, Aspergillus niger, Sclerotinia libertine, then lipase, Candida lipolytica, and cellulase, Trichoderma conigi. Name of the enzyme along with the microbial source is very important topic which is given on page 261. Microbes in sewage treatment. Sewage is a matter carried off in drainage. It is a municipal waste containing human excreta, household waste, dissolved organic matter, and even pathogenic microbes, that is bacteria, viruses, protozoans, nematodes, and microfungi, etc. It also may include discharge water from hospital waste, slaughter house waste, animal dung, etc. Discharge from industrial waste. It contains toxic organic and inorganic chemicals, tannery, pharmaceuticals waste, etc. Sewage treatment process includes 4 basic steps as follows. Preliminary treatment or primary treatment, and then secondary treatment or biological treatment. Primary treatment or preliminary treatment, it includes screening and grit chamber. You can see here in the nearby picture, grit chamber is shown along with sedimentation tank. So that is primary or preliminary treatment. It is a physical removal of particles. It includes removal of floating debris by sequential filtration, removal of grit by sedimentation. Secondary treatment, it is also called as biological treatment. The primary effluent is passed into large aeration tank. Here it is constantly agitated mechanically and air is pumped into it. Aerobic bacteria grow vigorously and form flocs. Flocs are the masses of bacteria held together by slime and fungal hyphae to form mesh-like masses. These aerobic microbes consume the major part of the organic matter present in the effluent. Now, due to this, BOD, that is biochemical oxygen demand of the effluent, is significantly reduced. Now, in secondary treatment, tertiary treatment also starts. That is, once the BOD of wastewater is reduced, it is passed into a settling tank. The effluent is then passed into a settling tank where the bacterial flocs are sediment. This sediment is called activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining sludge is pumped into large tanks called anaerobic sludge digester. Here some anaerobic bacteria digest the bacteria and fungi in the sludge by producing gases like methane, hydrogen sulfide and carbon dioxide. These gases form the biogas. So here you can see the production of biogas in the tertiary treatment. Microbes in production of biogas or microbes in energy generation. Very important diagram along with the topic also. Biogas is a mixture of gases, mainly methane, produced by the microbes such as methanogens. Methanogens grow anaerobically on cellulosic material and produce methane along with carbon dioxide and hydrogen. Methanobacterium is found in the anaerobic sludge and rumen of cattle for cellulose digestion. Biogas is used for cooking and lighting. Cattle dung or gobar contains these bacteria, so dung is used to produce biogas or gobar gas. Anaerobic digestion involves three processes like hydrolysis or solubilization. It is the initial stage. Raw material is mixed, that is cow dung or cattle dung is mixed with water in equal proportion to make slurry, which is then fed into the digester. Then acidogenesis. In this stage, facultative anaerobic acidogenic bacteria and obligate anaerobic organisms convert simple organic material into acids like formic acid, acetic acid, hydrogen, carbon dioxide, etc. And then the next stage is methanogenesis. This is the last stage in which methane gas is formed. Parts of biogas plant: a concrete tank, then outlet to supply biogas, then outlet to remove slurry. It can be used for the fertilizers or it can be used as a biofertilizer. Benefits of biogas production. You know that it is a cheap, safe and renewable source of energy. It can be easily generated, stored and transported. It can be used for domestic lighting, cooking, street lighting as well as small scale industries. It burns with blue flame and without smoke. It helps to improve sanitation of the surrounding. It is eco-friendly and does not cause any pollution. So, benefits of biogas are given on page number 264. Just go through the points. Role of microbes as biocontrol agents. Biocontrol is the use of biological methods for controlling plant diseases and pests. For example, ladybird controls aphids. Dragonflies control mosquitoes. Chemical pesticides and insecticides kill both useful and harmful organisms and cause pollution. Biocontrol method has no such problems. So biocontrol agents are pollution free. Microbial biocontrol agent. There are some examples. The first one is Bacillus thuringiensis. We have studied in 10th standard about Bt cotton and Bt brinjal. In that, bacterial or Bacillus thuringiensis is added. Bt, that is Bacillus thuringiensis, it is to control butterfly caterpillar. The dried spores of Bt are mixed with water and sprayed on to vulnerable plants such as brassica, fruit trees. These are eaten by the caterpillar. In their gut, the toxin is released and the larvae get killed. Scientists have introduced Bacillus thuringiensis toxin genes into plants, for example, Bt cotton, Bt brinjal, etc. The second example is Trichoderma species. They are seen in the root ecosystems. They control several plant pathogens. Let us now learn very important topic of the lesson, that is microbes as biofertilizers. You know that biofertilizers are organisms that enrich the nutrient quality of the soil. For example, bacteria, fungi, cyanobacteria, etc. We have studied about rhizobium, symbiotic bacteria in root nodules of leguminous plants. They fix atmospheric nitrogen and convert it into compounds of nitrogen, that is nitrates and nitrites. Free living bacteria in the soil, for example azospirillum and azotobacter, enrich the nitrogen content of the soil. Then there are phosphate solubilizing biofertilizers, for example pseudomonas bacteria, bacillus bacteria and agrobacterium. Then compost making biofertilizers. Then cyanobacteria as biofertilizers, for example Anabina, nostoc, azolla, etc. Most important topic, that is fungal biofertilizers. Fungal biofertilizers are also called as mycorrhiza. It is a fungus. It forms symbiotic association with underground parts like rhizomes and root of higher plant occurring in thick humid forest. There are two types of mycorrhizae, that is ectomycorrhizae and endomycorrhizae. Now ectomycorrhizae, they have well developed mycelium that forms mantle on the outside of the root. This increases absorptive surface area of roots and accelerates uptake of water and nutrients. Due to this, the plant vigor growth and yield increase. Some hyphae of mycorrhizal fungus penetrate into the root and forms heart's net. Endomycorrhizae, they grow in between and within the cortical cells of root. They are opposite to ectomycorrhizae. Ectomycorrhizae are present outside the root and endomycorrhizae are present in the cortical cells of the root, inside the root. Fungal hyphae penetrate the cells and form finally branch arbuscules and form vesicles, mostly in the intercellular spaces of cortical cells. Hence they are called VAM, that is vesiculo-arbuscular mycorrhizae. The full form is very important, VAM, that is vesiculo-arbuscular mycorrhizae. Biofertilizer microorganisms, rhizobium, already we have discussed, azotobacter. It is important and well known free living, nitrogen fixing, aerobic, non-photosynthetic, non-nodule forming bacterium intimately associated with roots of grasses and certain plants. It is used as a biofertilizer for all non-leguminous plants. Then azospirillum, it is free living aerobic nitrogen fixing bacterium associated with root of corn, wheat and jawar. Then Anabina, it is a genus of multicellular filamentous cyanobacteria that exists as plankton. It has ability to fix nitrogen and also form symbiotic relationship with certain plants such as the coralloid roots of Cycas and Anthoceros thallus. And the last is azolla. Azolla is a free floating water fern. Azolla plant consists of a floating rhizome with small overlapping bilobed leaves and roots. The leaf shows dorsal and ventral lobe. So here we have finished with the lesson microbes in human welfare, enhancement of food production. Thank you and have a nice day.